So, in my lab, we are committed to understanding how the human immune system functions in a healthy way in response to infection, and then also under circumstances where it dysfunctions and causes disease, like during chronic infections, where the immune system itself can actually mediate the damage. And human immunity, I suppose, is fascinating to me because it's so integral to human health. It has a role to play in all parts of your body and in every illness. And I'm especially interested in human immune responses in the lung. And we've been studying ways to boost and support immune responses against infection, and also ways to dampen down and regulate inflammation when it gets out of hand, because the lung is a very delicate environment. With COVID-19 vaccine development taking centre stage for so long, we took the opportunity to assess how certain human innate immune function can be altered by vaccination with different types of vaccine platforms, such as the more traditional adenoviral vectored vaccines versus the newer mRNA platforms. And vaccine effectiveness, of course, is traditionally measured by adaptive immune responses, which confer protection against the target bug. But real world evidence actually indicates that certain vaccines are capable of much more than that and can have health benefits outside of their direct target effects. And that's really what we're interested in. From animal models using adenoviral vector vaccine platforms that a phenomenon known as trained innate immunity occurs in mice, whereby the adenovirus vector actually makes them better able to fight off bacterial infections. And then more recently, a review of the COVID-19 human vaccine trials indicated that adenoviral vector vaccines can protect against all-cause mortality and significantly reduces non-COVID, non-accident deaths. And the molecular mechanisms behind this observation are really completely unknown. And this was absolutely fascinating to my team. And one of my very brilliant PhD students, Derv Murphy, hypothesized that the Oxford AstraZeneca vaccine that uses the adenoviral vector can be perhaps is reprogramming innate immune function and training these cells then to be better fighters of infection in general. So we're now continuing this work to help us to understand how this innate immune reprogramming can alter adaptive immune responses, with the long-term aim of using this evidence to help to identify the best vaccine platform for use against new infections and to optimize prime boost regimens and limit serious side effects. And our work really improves the understanding of the contributions of innate immune function to these specific and non-specific vaccine effects, and it will hopefully enable us then to innovate around the design of new vaccines for very difficult to treat infections like tuberculosis and new and emerging threats to global health.